Hello, everyone, and welcome to part 2 of our webinar series on 3D culture and analysis, getting started in 3D, setting up your lab and reviewing common pitfalls of researchers new to 3D. I'm Alexis Cross of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question, and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credit. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Dr. Mark Kennedy, R&D scientist at Thermo Fisher Scientific Cell Biology 3D Models. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Mark, you may now begin your presentation.
Thank you for that introduction, and welcome everyone who's taken the time to join us here today for this presentation titled Getting Started in 3D. And so my goal with this presentation is really aimed at people or scientists who are new to working with 3D cell models, or people who perhaps have not yet decided to start working with 3D models, but are very much interested in doing so, and are just looking for some information on practical considerations for moving into this new area of cell culture models. So just briefly, what I plan to cover, we'll start with just a very light introduction to 3D cell models. I'm also going to talk a little bit about some of the common reagents that are used, as well as point you towards some of the available resources that exist on the Thermo Fisher website or elsewhere that really may help point you in the direction of identifying what it is you need in order to get started, or to help you choose what model might be best for answering the questions that you're most interested in addressing. After that, I'm going to talk a little bit about a couple of it says here common techniques, but perhaps it should be uncommon techniques. Just some information about some differences that you will very quickly run into, some of which that are probably self-evident. But regardless, if your background has been primarily or only working with traditional 2D monolayer cultures, there are some, not subtle, but there are some things that you will run into quickly that may present challenges to newcomers to 3D cell models. And lastly, we'll just talk a little bit about some of the other sort of almost advice points or tips and tricks and things to be aware of for people who are interested in exploring or moving into more of a 3D cell culture workflow. So with that, I would like to start by just briefly mentioning or introducing people to the wonderful world of 3D cell models. As many of you, I'm sure, are aware, this is an emerging field right now that is really drawing a lot of interest across a wide range of research areas. It's really been climbing into prominence over the past decade or so, with the publication of several key or seminal findings, only just a couple of which are highlighted here in the graph on the left. And these represent some of the foundational pluripotent stem cell derived or adult stem cell derived organoid models that are really helping propel 3D models forward. And as you can see here, this graph is a representation of the number of publications being published year over year for the past couple of decades. And you can really see here that really driving the interest here, there's people from all areas of cell biology are taking advantage of the new things that 3D models has to offer. To sort of break that down a little bit, on the right side of the screen here, of the slide, this is a simple breakdown of this PubMed search, just to give you an idea of the types of tissue models or disease models that are most heavily, where researchers are most heavily using different types of 3D models. Cancer today appears to be probably the field where 3D models are having the largest impact early on. However, there are many other tissue types in which 3D models are allowing scientists to ask new questions about development and disease in a way that they've not been able to do before. And so really the use of 3D cell models is becoming quite ubiquitous across all walks of research. So when we say a 3D cell model, at least for the purposes of today's talk, what we're referring to really is a cell model in which you've got an aggregate of cells. So as opposed to traditional monolayer culture where cells are grown sort of, well, in monolayer on some sort of flat surface, the 3D models tend to take advantage of aggregating cells in one format or another, or using bioprinting, for example, to create complex three-dimensional structures from cells that allow them to make these advanced cell culture models. And somewhere in between the two are some various co-culture models, as well as the microphysiological systems or the organ-on-a-chip type approaches, where in some instances these may appear more like a 3D cell models. In other instances, they're a little bit more like it's tissue engineering of a 2D monolayer. But regardless, these all represent some of the different flavors of 3D cell models that are growing in popularity. And for today's talk, the emphasis of the information that I'm going to give, although it's not limited to spheroids and organoids, that will represent the 3D models of focus. So for most of what I'm talking about, and I'll refer to them as we go, usually I'm going to be talking about spheroids and organoids as a 3D model. And one of the places where I'd like to start is with some definitions. So this is an area where definitions are still somewhat unsettled in the field. And you may have yourself differing definitions here, but again, for the purposes of today's talk, so that when I say a spheroid or an organoid, you understand what I mean, even if your definition doesn't fully align, because there are areas in which this terminology really becomes a little bit hard to follow sometimes. But for our purposes anyway, a spheroid is really just an aggregate, a 3D aggregate of a cell line or primary cells that self-aggregates into what almost looks like a ball of cells, really. And these spheroids, the cells in these spheroids are able to grow and proliferate, and they may display some altered or enhanced physiological responses compared to their 2D counterparts. However, kind of what you put into this model is all that's ever in there. The cells may grow and proliferate, but there's no differentiation or change of cell types that really happens. So in other words, there's really no stem cell compartment to these models. However, when I talk organoids, what I'm really talking about is a stem cell derived model. And so these can be models that are derived from either pluripotent stem cells, such as embryonic stem cells or induced pluripotent stem cells, or they can be cells isolated from tissue, adult or embryonic tissue, in which a tissue specific or tissue-resident population of progenitor stem cells are able to self-organize into little microtissues that resemble the organ from which the cells were originally isolated. So, and this is in line with some published definitions around organoids, and this is a snapshot of one here that we like to use for way of example. So there are many reasons why researchers are moving into 3D cell models. This table is just meant to capture a few of those reasons. It's becoming more and more recognized that although 2D models have been and will continue to be very useful for certain applications, they are very simplistic, and as it relates to drug discovery, for example, the oversimplicity of these models is often cited as one of the reasons why traditional 2D models are not very predictive of a cell's response to a given toxicant or some form of compound. And so 3D cell models, however, appear to be giving a more in vivo-like response in many cases, and in some reports seem to recapitulate more of or behave more in line with clinically relevant data where available. So some of the considerations here between 2D and 3D cell systems that may help one decide which model is right for their question is looking at some of the things like the culture conditions, for example. Conveniently, in 2D monolayer culture, cells are uniformly exposed to the cell culture media, which means anything that you put in the media, all cells are more or less uniformly exposed to them. Whereas in 3D systems, really only the cells on the outside of the 3D model is exposed to the full concentration of any given compound. And so this sets up diffusion gradients across the 3D microtissue, and this has impacts for its biology. And we'll come back towards the end of this talk with another note about that. Some of the other things that you may be aware of, or that is good to be aware of, is that 2D systems, oftentimes the gene expression or the protein expression signatures of these cells don't correlate terribly well with the signatures of the tissue from which they were originally isolated. Or in the case of a patient-derived cancer cell, they don't always, or in fact usually they don't look much like the tissue or the cancer or the tissue of origin. One of the nice things with 3D cell models is that they seem to hold the gene expression signatures or protein expression signatures much more faithfully, such that they look more like the in vivo tissue or disease sample compared to 2D systems. However, although this sort of enhanced biology, if we can call it that, is a great advantage, there are other things that sort of are standing in the way or are currently limiting 3D systems in terms of their more widespread adoption and the ability to scale up some of these models for more high throughput workflows. And that leads to things like the lack of validated assays for 3D workflows. While many assays can be sort of co-opted for use in 3D, there tends to be a lot of optimization that one has to use now, since most of the available assays were developed for 2D cultures. And so there is some consideration that one has to take there for optimizing assays. And it also, because they're 3D, it tends to be a little bit of or somewhat of a low throughput for various types of imaging analysis, for example, that may require cryosectioning or paraffin sectioning as opposed to simply labeling the entire monolayer culture. So there are challenges like that new scientists to the field should be aware of. And this kind of relates also to the complexity of the model, in that although 3D models are more complex than normal 2D models, this also makes them somewhat technically challenging to grow, to handle, and to assay. The good news is that all 3D cell models start with the same foundational pieces that all cell culture models start with. That is, they require cells, they require some sort of vessel or substrate to grow on or in, and they all require some cocktail of media and or supplements and growth factors or the like in order to grow and proliferate or potentially differentiate and form these different tissue-specific organoid models. So just for point of reference here, some ways of example of this is that cells that are commonly used today, a lot of them are stem cells, either pluripotent stem cells or various iPSC cells that have been reprogrammed from patients, for example, using patient-derived cells, as well as various cancer cell lines. But there's also a lot of people using various types of primary cells, such as primary human hepatocytes, and I'll come to this, I think, on the next slide as well. Many of the culture vessels and matrices that you're already familiar with also are used in these workflows. These include matrices such as Geltrex, which is essentially a basement membrane-like extract that's used as an extracellular matrix mimic. Or there are other scaffolds, such as the Algematrix matrix that Gibco currently offers, and as well as other things like collagen. There are also many models require the use of plastics that prevent cell attachment so that the cells basically grow in suspension. And the use of these super low attachment plates, such as the Nunculon Sphera microplates and dishes and flasks, are also very useful in culturing various types of 3D models. And like I said, many of the media and supplements that I'm sure many of you are already aware of, things like the StemFlex media or the Essential 8 media for culturing robust stem cell cultures, and other common or popular basal cell media formulations such as the RPMI 1640 media, advanced DMMF12 medium, and various others. And supplements like B27 and various flavors of B27, and of course lots of different growth factors. The other things that can be used for analyzing these systems then are things like high content analysis platforms, as shown here on the top right. But you can also do more standard type approaches as well, where these cells can be taken, RNA isolated for doing qPCR type analysis of gene expression signatures, or of course cells can be isolated, and other more sequencing type technologies can be applied to better characterize the cell population, and also using sort of more standard upright fluorescent microscopes that are more benchtop-like instruments, such as the EVO cell imaging systems. And again, many of these can be interrogated with a lot of the reagents. At least looking at cell health can be interrogated looking at a lot of the reagents that are commonly available through molecular probes, as well as various antibodies and labeling kits, or the use of TACMAN assays, for example, to really characterize what you have in your 3D cell model system, or if there's an alteration in the cell health of your model, you can also monitor that reasonably effectively as well with different types of cell health or viability kits that are commonly available. As I mentioned, there are a lot of the cells that are currently in use today in 3D cell models include things like primary cells. So these include things like endothelial cells, which may be used for modeling vasculature, but also primary human hepatocytes and other animal hepatocytes as well, where these, under non-adherent conditions, can also form aggregates or spheroid-like cultures, and this provides some advantages that 2D cell cultures simply don't have. For example, with respect to hepatocytes, one of the traditional challenges with working with primary human hepatocytes is their longevity in culture. So in typical 2D cell culture, a good lot of primary hepatocytes only lasts for probably somewhere around 4 to 6 days. A really good lot, or with some special media that are available, that might be extendable out to more of 8-10 days or so. But simply culturing hepatocytes in a spheroid culture immediately provides huge advantages to the longevity of this model, as it's been shown by scientists at Thermo Fisher, as well as various other places, that spheroids or hepatocyte spheroids can be cultured for many days and many weeks, allowing scientists to study other features of drug toxicity, low turnover compounds, and things that weren't very easily achievable before 3D. And of course, there's the widespread use of stem cells such as iPSCs, which can be gene edited to make disease models, or if they're patient derived, they could be used to make isogenic lines for pairing controls and disease models for studying disease etiology in a 3D cell model. So this table is also just meant to summarize some of, certainly not all of the landmark publications across a number of different organoid model systems. And again, just to orient you to the fact that many of the cell culture ingredients that you may have been using for other purposes have often been directly co-opted for many 3D model systems. So these include many different basal media, like I alluded to earlier. Advanced DMMF12 is certainly a very common media used across the adult organoid model systems, as well as neurobasal and traditional DMMF12 and various other iPSC-derived models. And there's a whole host of different recombinant factors or growth factors that can be used to help direct the differentiation of these pluripotent stem cells so that you create the organoid model that you're actually interested in making so that it contains the cell types that are relevant to you. And many of these are currently available today. One of the things that we hear a lot, especially for people who are just getting started, is again looking for resources to help identify what reagents, help them plan, since these are tend to be very long experiments. Having the availability of these resources is very important for most people that we talk to. And this is an example here. You can go to the thermofisher.com/3D models website for more information and for all of the things listed here. This is just meant to be a highlight of the website or of information from the website, just to let you know that there's lots of information on this website and also in many other places that you can find with a simple Google search to help you identify protocols and the methods that may be used, as well as if they currently exist in Thermo Fisher, for example, if these are existing products. It's organized in a way where things are easy to find to help get you started. There's also technical resources that are available freely. These include things like application notes and scientific posters to help show you how internally in R&D we've been using various reagents in different 3D cell model systems. And in some instances, there are also some how-to videos that we've put up from collaborators to help people get started in certain model systems. And this website is by no means sort of one and done. It's certainly something that is being continually updated. So if you are choosing to work in 3D cell models, remember to periodically check back as there may be new information relevant to what you're interested in studying. And along these lines, just a couple of last things here. Other resources that are available for people to download are things like we have an ebook centered on cell culture and analysis, as well as the standard molecular probes handbook, which contains many of the different molecular probes that can be used to assay various features of cell health. And there's also a very nice e-learning course that you can freely register for, and there are, I believe, a bunch of modules in this to sort of take you through a bit of the history of 3D cell models and some information on different types of 3D cell models that you may find just very informative. So, with that in mind, one of the things that I'd like to briefly start with is talking about models or organoid models in particular that start from different types of stem cells. And one of the features that is also self-evident here is that the quality of the material you start with is extremely important. And and you may hear phrases such as garbage in, garbage out, and I think that really applies to these models, in part because they do tend to be very long protocols. And so if you start with stem cells that are poorly characterized or growing poorly or have some routine culture issues, you really are setting yourself up for failure. And so in view of that, I think it's really good practice to remind oneself to really take the time and periodically characterize your cells, make sure that they are of sufficient quality to make it worth the investment to move into a 3D cell culture model, because these do tend to be reagent intensive protocols. So one of the things that's useful to do, shown here across the top, and this applies to pluripotent stem cells as well, which are cartooned on the bottom left. But if you're working with stem cells, it is good to periodically characterize the chromosome number or chromosome abnormalities to get a sense for what the genomic stability of your system is, that there aren't some chromosomal abnormalities that may be contributing to aberrant differentiation methods, so that when someone else tries to use a given method on their own cell line, they get different information than what was published. So it's really important to characterize your cells, the genomic stability of your cells. This can be done doing things like karyostat. You can also do various techniques for studying copy number variation, just to get us, and also somatic mutation rates, just to make sure, once you've taken your cells into a culture system, that these models remain fairly stable and don't start accumulating rates that are sort of more common than they are in other 2D cell culture systems. And of course, doing regular mycoplasma testing as well is sort of a standard in cell culture, but also important to remember to do. And as it relates to pluripotent stem cells, checking to make sure the cells really are, your population of cells really are pluripotent is very important, so that you don't have some colonies of differentiated cells mixed in with your undifferentiated cells that may make it difficult to reproducibly generate the iPSC model that you're interested in, so that, in other words, you don't have competing cell populations or populations of cells that may influence your model in a non-desired way. And so monitoring pluripotency of pluripotent stem cells can be done in any number of ways, including using commercial tests such as PluriTest, or the TACMAN-based TACMAN scorecard system. Can also be done by looking at immunocytochemistry of various pluripotency markers such as OCT4 and NANOG and others, to really get a good sense and make sure the bulk of your colonies are indeed in the differentiation state or undifferentiated state that you want them to be in before starting your experiment. Some of the other things to be aware of, and I alluded to this a little earlier on, is that these cell models are three-dimensional. They're not really in the appearance of in sort of an X and Y kind of arrangement. There really is depth to these models, and so the thickness of these models also creates gradients of various types across the microtissue. This includes things such as gradients of oxygen and nutrients and growth factors that are in the cell culture media. And so in a 3D cell culture model, these are the cells on the outside of your spheroid or your organoid. See the highest concentrations of these things. And as these molecules, or as you look across the diameter of these models, the exposure of cells towards the center to oxygen and nutrients decreases. And this, of course, sets up somewhat of a counter gradient of carbon dioxide and various other metabolic waste products that will accumulate then more towards the center of your model. And in some cases, when a model becomes too large, this creates or results in the creation of what people usually recall a necrotic core, where the center of your organoid or your spheroid is essentially just dead tissue or dead cells. Other things to remember, drug exposure would follow the same thing. Cells on the outside of your model system certainly exposed to higher concentrations of a given compound, whereas cells towards the middle tend to be exposed to slightly different concentrations. And for people studying toxicology, this can result in differences in the types of IC50s or EC50s that you may be looking to characterize for a given compound. And in some reports, this is thought to be somewhat of a good thing, because in vivo, all cells are not uniformly exposed to compounds in the same way and at the same concentration. So in some ways, this gradient is meant to mimic in vivo a little better there as well. In addition to this, there are other features to be aware of. So I mentioned the necrotic core, and on the very opposite of that, so on the very outer region of a spheroid model, is where you tend to have a proliferation zone. So cells on the outside tend to be more proliferative than cells on the inside, which tend to be a little bit dead once a spheroid reaches a certain diameter. For a lot of cell lines, that tends to be in the 250 to 300 micrometer diameter range. And as well, there's sort of a middle layer of cells which are not really proliferative, but they're certainly viable and metabolically active. And so just to give you a real-life example here of a necrotic core, this is a spheroid grown from HepG2 cells, which are a hepatic carcinoma cell line. And you can see here, these are labeled with a molecular probe called cell event caspase 37, which lights up apoptotic cells. And you can see here a concentration of these dead and dying cells in the center of this spheroid. Other practical considerations that you may be aware of, but is important not to underestimate, is the method of aggregating your cells in order to make a 3D cell model system. Some of these can be thought of as either specialty plastics or perhaps non-standard platforms in some instances. And in other cases, they can just be kind of cumbersome, although effective, but cumbersome kind of approaches to aggregating cells. So on the top left here is an example of what we call the hanging drop method, where literally you put a fixed number of cells in a small droplet of media on a plastic surface, and you turn that upside down so that it creates a hanging drop. And this results in the sort of aggregation of the cells toward the apex of the drop, where they are able to self-aggregate into a spheroid. This can also be achieved somewhat more easily in methods such as on the right here, where this is meant to be an illustration of a single well in a 96 well U-bottom super or non-adherent plate. So these are plates in which the inner surface of the culture well has been coated or treated in some way that prevents cell attachment. And so when you put your cells in, they all aggregate to the bottom, and the shape of the bottom, or the shape of the well at the bottom, forces all the cells to kind of aggregate together more quickly. And again, this often, for many cell lines and cell types, this results in the formation of a spheroid. Other types of formats that are used to make or to aggregate cells, either cell lines or pluripotent stem cells, or also for long-term culture of them, are things like mini bioreactors or spinner flasks. These are things that you may not have as a standard cell culture vessel in your cell culture hood. I'm sure most people are probably more adept at simply moving plates that all look the same with different numbers of wells or dishes of slightly different diameters in and out of their incubators. But mini bioreactors or spinner flasks, they use a magnetic impeller, which means they require a magnetic plate or a magnetic stir plate to turn the impeller to help sort of move the media around. And some of the reasons why this is done is that while it may also help improve spheroid formation as well as consistency in the size of the spheroid, it also, the agitation is also thought to help better feed some of these spheroids. So trying to mitigate the effect of growing too large and forming necrotic cores, which may not be desirable for many cell models. It provides advantages there. And then somewhat comparable to that, although less commonly seen, would be the use of roller bottle cultures, which would effectively be doing the same thing, but probably just a less common approach in the literature from what I've seen. So in keeping with some considerations for spheroid growth, if you're interested in making spheroids from cell lines or various types of primary cells, typically you don't need any special media. Using what you've already been using, usually some cocktail of DMEM or DMEM-F12 with some fetal bovine serum, reagents that reduce contamination, such as Pen-Strep additives or anti-anti or primicin or other types of reagents to help reduce contamination issues, and supplements, Glutamax, things that the cells like. And growing these in one of the formats as shown on the previous slide is really all you need to do to get started for many cell lines. And also simply for aggregating, for growing many cell lines. It gets a little more complicated when you're working with pluripotent stem cells. The media may become a little more specialized. The use of growth factors, specific growth factors, may become a little more, may become a little different. But otherwise, this is kind of the basic setup here. So this is meant to illustrate sort of the last line with that is that traditionally, I know when I first started culturing stem cells or to form embryoid bodies from stem cells many years ago, one of the ways in which it was commonly done, partly because it was cheap and somewhat effective, was simply to culture cells in static culture, but in vessels that were non-adherent, so that cells would not attach. And over the course of a couple of days, cells would either aggregate and form, or simply through cell proliferation would form spheroids, but they would form spheroids of many different sizes. And the size of the spheroid can impact how well it differentiates and the health of spheroid A versus spheroid B if they're of different sizes. These are things to keep in mind. And so some of this can be controlled through the use of formats such as U-bottom, super low attachment plates or spheroplates, in which a fixed number of cells can be put into each well so that it forms a spheroid of more or less a known size in a fixed period of time. And so the graph on the bottom right here is just meant to illustrate the consistency between 8 replicates of spheroids formed from 4 different initial seeding densities, and just to show that consistency in spheroid size from spheroid to spheroid is quite consistent, and the size of the overall spheroid is directly proportional to the number of cells that you start with. And so again, these are important considerations for folks who are just getting started. One of the other things to keep in mind, and I know speaking from personal experience, when I first started working in 96 well U-bottom plates, was a little bit of an afterthought when I was getting started as to how was I going to change the media. And this is important because these are essentially suspension cultures. So as we said, they form aggregates form because the internal surface of the plates are treated so that cells don't attach. And so this means if you want to aspirate your media, you're also going to aspirate your cells. And so there are several ways to sort of minimize this risk. One way that I find useful is to use multi-channel pipettes. Of course, if you're working in 96-well format, you're probably working with multi-channel pipettes anyway. But you can manually do media changes. Usually it's done by not trying to remove the entirety of the media, but perhaps doing successive 50 or 75% of the volume, removing that and adding it back to sort of dilute out the spent media until you just have fresh media. So this is an effective way. However, it still comes with some issues of being consistent and being able to hold a a multi-channel pipette level for a fairly long period of time, which sounds trivial, but in practice is not always. And so you can end up with inconsistencies in volume across the plate. So although it is effective, it's still, it can be a bit tedious and slow, and it's certainly not without some limitations. Another way around this has been the use of plate washer systems. So it's a way of basically automating your spheroid media changes. So microplate washers, such as the well-wash Versa, has controllable settings so that you can control the position of the aspirator nozzle, as well as the speed of the aspiration and the height of the nozzle within the well, so that you can essentially program this thing to aspirate your media without losing any of your spheroids. And if you like it, of course, it can add your media back, or if you want, you can do that part by hand, depending on the number of plates you have. It may or may not be needed, but it's certainly a much more efficient, fast, and in our hands has been a very reliable way of very quickly aspirating a fixed volume of cells across, a number of 96-well plates, and with a level of consistency that we're just not able to do with multi-channel pipettes very easily. So another thing to be aware of is that many of the 3D cell culture models, especially on the organoid side of the 3D cell model space, is the use of extracellular mimics or matrices for growing these models. And so these are used to sort of encase the cell aggregates in order to help promote further maturation or some of the structural organization that people have reported and are commonly seen in various types of organoid models. Culturing, the ECM is a very important component of that system. So using materials like Matrigel or Geltrex, which contain many of the components that are normally found in extracellular matrix or matrices, are really useful in helping promote various types of model systems. However, these materials are temperature sensitive, meaning you have to be careful, you have to work with them under conditions that are sometimes hard to keep sterile, so keeping things on ice, for example, so that the ECM mimic doesn't polymerize while you're working with it, so trying to keep your materials cold while you might want to keep your plate pre-warmed, so that as soon as you deposit Matrigel or Geltrex onto the surface, it helps promote the polymerization quickly, so that the droplet or the dome of Matrigel in which you resuspended cells, polymerizes quickly, so that it doesn't lose its shape and basically just become trapped in what looks like a coating on your plate. And so this is commonly used for growing certain cell lines, as well as patient-derived cells, and various types of adult stem cell organoid models. And just by way of example here, so this picture on the top left is an example of 3 domes of an extracellular matrix mimic in which cells have been reconstituted so that they can grow into spheroids, either multiple spheroids or one spheroid, depending on the model system and what the cells were like before you resuspended them in the matrix. One other thing that's important to point out is that these materials are also quite viscous, and so in order to pipette accurately with these materials, it's definitely a good idea to work with positive displacement pipettes instead of the air displacement pipettes, which are probably a little more standard in most cell biology or molecular biology labs. It certainly plays dividends in pipetting the exact same volume from droplet to droplet, which can impact how these things are being fed. So adult stem cells are grown in this way, where cells isolated from tissue are resuspended or reconstituted inside of an extracellular matrix. And many of them grow into what almost looks like bubbles inside the gel. So they form these cystic-like structures, but if you actually stain them with something such as DAPI, you can see that there are these cellular models, these are lighting up all the different nuclei representing all the different cells in a given model shown here on the right. But this really only works if you're growing cells inside of an ECM. Another way in which ECM is used, which is maybe a little more common or commonly seen in the pluripotent stem cell space, and that is to either to take aggregates or previously formed aggregates of cells and to put them inside droplets of matrigel or Geltrex. And so the red arrow here in the picture on the bottom is actually showing a spheroid or a growing organoid, I should say, that is differentiating into certain lineages. And at a certain time point, we have to work very quickly and keeping the temperature sensitivity of the material in mind, try and dispense or deposit spheroids inside of droplets and position them so that when the gel polymerizes, they're fully encased, so that they can then go on to grow into whatever model system you're looking at. The picture shown here on the bottom right is just a picture of a neural organoid, and you can kind of see around the edges here a lot of the almost like folded epithelial-like structures. Although the center is pretty dark, suggesting that the center probably will have a lot of dead cells in it, and this is not too uncommon, and certainly something to keep an eye on when you're monitoring the health of your system. So as I wind down here in the last couple of slides, a couple of the other things that are probably relevant or important for people who are looking to get started in 3D or who have only recently started in 3D is that these model systems tend to take very long to grow. So some of the cell line model systems are probably much shorter. They would grow on the order of days to maybe a week or two, depending on what model system you're using and how large you're trying to have it grow to. But typically in the organoid space, protocols last anywhere from several weeks, potentially to many months before they're actually sort of at an endpoint where they're useful for assay or potentially useful for assaying. And so this is done to help promote the maturation of different cell types, especially if you're talking about pluripotent stem cells. But the take home really is that because these are really long protocols, it's really important to plan ahead to make sure you have everything you need to sustain your culture through the term of the experiment, so that you don't sort of go, oh, I've been culturing for a month, I've got one more month to go, and you find out you're in between orders, and suddenly you might not have material for the next media change. Although that sounds a little bit pandering, it is really not meant to. It's just meant to emphasize that in these long protocols, I mean, planning as always is really important, but really planning maybe a little longer term than one may be a little more accustomed to doing. Another part of this also has to do with what we called earlier the less common culture formats. The use of roller bottles or mini bioreactors or spinner flasks, or the use of orbital shakers. Maybe not always in the form of flasks like shown here. Sometimes they're orbital shakers that you would put in your incubator, on which it's a flat platform, so you can lay various types of dishes in there for agitation to help with the feeding and long-term growth of your organoid model system. So the reason why this is certainly something to plan for is because these things take up different amounts of space in a typical cell culture incubator than simply putting plates on a shelf will typically do. So the space required to grow these is certainly something different that definitely requires planning for, especially if this is not the only thing your lab is doing. You want to maintain space for other experiments that maybe aren't using these sorts of less common culture formats. It does require planning ahead and, of course, working with your colleagues. The other important feature to remember here with with respect to the length of these cultures is that these really long or extended culture times, it really enhances or compounds the risk of contamination. And I know in talking to lots of customers and collaborators who work in various types of models, although I'm sure people don't always like to admit to it, the longer you are doing something in cell culture, you're taking things out of the incubator many, many times, your risk of contamination just goes up. And when you're investing the amount of time it takes to grow an organoid, for example, to a useful endpoint, and considering the investment of resources needed to grow these models, contamination is something that one should take extremely seriously, and really maybe take extra precautions to guard against, because if you go 3 months into a protocol and it gets contaminated, you kind of have to start over. So it's definitely something to keep on top of mind. And the other piece, I guess that's important to call out with respect to the length of culture is that, and with respect to planning, is that doing iterative analysis on a given system when the protocol is long also means your entire study is going to take a lot of time. And so with that in mind, remembering, maybe staggering cultures might be one way to do it, so that you've got multiple experiments going that are just delayed by a fixed time, so that you've got multiple repeats that are sort of coming to an end at various times, so that you're not starting from scratch every time you need new samples for analysis. And so, although some of this may sound a little bit doom and gloom, it really should not, because these model systems are really interesting and are allowing people to study things in a way they haven't done before. But it is also important to remember to choose the best system, or the system that's best suited to your needs. And it's important to ask yourself, how complex of a model do you really need? So it may be that in some cases, the traditional 2D model system will still get you to the endpoint where you would like to go. It may be that you need a 3D cell model system. And then with respect to 3D models, there are considerations to be had with, well, how many cell types do I really need in this model? Do I need a neural organoid that contains or models as much of the brain as possible? Or maybe I just need a region-specific model system that might be a little easier to grow, a little more controlled, and get you to the endpoint where you want to go. And also considerations, between adult model systems and pluripotency or PSC-derived model systems. The level of maturation of cells may be, something that one has to reckon with and decide, how mature is enough for your study, and those sorts of considerations. And so with that, I really just want to end with a very brief summary. So again, 3D cell models are very exciting, and they're growing in population and popularity for a very good reason. However, they are very reagent intensive, and as such, they do tend to be much more expensive than traditional 2D cultures, and they do come with long and somewhat complex workflows. And so all of this kind of culminates in the idea that it's really important to choose the right model for what it is you're interested in studying. But again, the reason why people and why a lot of cell biology is moving toward 3D cell models is the desire to generate better models in order to generate better data as it relates to studying whether it be human development, disease etiology, predicting drug responses, identifying new therapeutics, using 3D cell model systems in any one of those ways in either regenerative medicine or perhaps in precision medicine. 3D cell models really are an exciting new tool in the cell biologist toolkit for addressing many of these questions. And so with that, I'd like to thank you all for your attention, and I'd like to close with a reminder that we are now able to open it up live for questions and answers. Thank you.
Thank you, Mark, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question, and click on the Send button. Mark will answer as many questions as time permits. So let's get started. The first question is, what are some of the differences between pluripotent stem cell models and adult tissue-derived models?
This is a good question. So pluripotent stem cell models obviously are made from embryonic or iPSC cells, and these are different from the adult tissue-derived models in that these cells have a broader differentiation capacity, meaning they can give rise to a wider variety of cell types given the right signals and culture environment than the adult tissue-derived models can. Where these are more restricted to cell fates that are typically found in the organ type from which or tissue type from which these progenitor cells were derived. And even more so, most of these models tend to only recapitulate the epithelial compartment of a given organ system. So what that means then is that they tend to have fewer cell types. Now, it is possible in some situations if you configure the right growth media to add in some of the missing cell types. But the pluripotent stem cell models are sort of able to generate more of these cell types perhaps simultaneously given the right culture conditions. However, pluripotent stem cells also are still plagued, or the field in general probably, are still plagued with the ability to generate a fully mature, fully functional cell from a pluripotent stem cell. Quite frequently, people will relate to how there's still a fetal characteristic to the cell as a way of relaying, that they're still not fully mature. And so pluripotent cell-derived 3D models, have that same issue. However, putting them into 3D is reported to help speed up or help improve the level of maturity and function that these cells can achieve in vitro, at least within a relative amount of time. Extended cell cultures may also help more or better mature these cells, but we're not always have the ability to culture these for 6, 8, 9, 10 months at a time for that to happen. Adult tissue-derived models tend to be more mature because they're already derived from an adult tissue. So it seems to be that they've already, through the course of normal development, have already passed through all these stages and have progressed into, an adult tissue cell. And so these seem to do a better job at then generating complex models that have more mature cell types.
Now, Mark, can you expand a little on the challenges and limitations of using matrigel or similar technologies?
Sure. So as I mentioned during the talk, a number of 3D cell models require the use of an extracellular matrix mimic or mimetic. And a lot of people, or a lot of these methods were built using a basement membrane extract called matrigel. And there are other similar technologies that exist out there. Geltrex and Cultrex are some of the other ones that are useful in making these organoid models in particular, but also some spheroid models. So some of the limitations to these materials is that they are temperature sensitive. Once they start to warm up, they start to gel or polymerize fairly quickly, which means working with them takes, you have to take extra care to be able to work with them. And so, working with cells on ice in a flow hood and those sorts of things just creates a little bit of an inconvenience, really, and may add some risk of contamination or add some contamination risks that one would want to be fully aware of. So, these sorts of problems are some of the challenges. In addition to that, these materials, they are derived from a mouse sarcoma. So I know frequently people question how relevant of a material it is, given that the composition of the material can vary from lot to lot to the extent that it requires lot testing for one's experiments, so that they could then procure, a relatively large amount from a same lot that functions for their needs, in order to be able to reproducibly generate their data. So these sort of undefined components or sometimes inconsistent components of these materials are certainly a limitation of it. And although you can probably control the stiffness of the material, which is a feature that I know some folks are interested in terms of trying to recreate an extracellular environment in vitro that looks more like the ECM environment in both its biochemical and biophysical properties in the organ in vivo. So although you can do probably some tuning of stiffness of these materials, being able to modify them with other peptides and also retrieving cells from these materials is not a trivial exercise.
How do you harvest or transfer spheroids from a 96-well U-bottom plate?
The short answer is very carefully. So depending on how big your spheroid gets, it'd be pretty tedious to do this one well at a time, but some handheld things like disposable sterile transfer pipettes can be done to do that. I tend to use the multi-channel pipette the same way if I'm using it for doing media changes, as I described earlier, where using a multi-channel pipette to remove just maybe 50% or 75% of the media and allowing the spheroid to stay on the bottom. The difference would be for transferring the spheroids was I would use a pipette or a multi-channel, preferably with wide bore tips, so that the spheroids can be aspirated up into the tip more efficiently, and then transferred to some other vessel for downstream manipulations.
And it looks like we have time for one more question. How do I know if the spheroid or organoid I made is any good?
That's also a very good question, and one that doesn't have an easy answer. At the end of the day, it's really going to require sort of testing to find out, one, what's how healthy is the sample I made? Do I just have a ball of mostly dead cells? And what impact is that going to have on my model? Is it big enough? Is it too large? Does it have the cell types that are relevant? There's just unfortunately, sort of no standard way of knowing what, if it's exactly what you need in advance of doing that sort of testing. But what one should probably do, and again, this is probably somewhat obvious, but given the length of these protocols and the increased amount of reagents that are usually required to generate these, it's probably good practice to try and identify some quality control steps in your method, so that you can routinely sample, acquire a sample of your spheroids or organoids, quickly monitor them by either imaging for their proportion of dead cells, or maybe doing a qPCR panel of a few number of genes to make sure your cell types are represented, or if there are functional markers that you can identify in that way to make sure that they are expressed at the time points in the protocol when you would anticipate them being expressed or being present. Really, it's just a way of trying to identify as early as possible in your protocol whether or not things are progressing in the manner that you want them to progress in, so that, one, you either continue to culture, or if you look at your gene expression and you find you have, what you feel is an unacceptable level of off-target differentiation into cell types that are not important for your model, then you've probably reached a decision point where you can stop and start over and try again.
I would like to once again thank Dr. Mark Kennedy for his presentation. I'd also like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We would like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through September of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be directed to register for Part 3 on 3D culture and analysis. Until next time, goodbye.